use them to swim. In our bodies, they're only used to move things along their surface. So you'll have a whole sheet of cells that has cilia on their surface. For example, in the respiratory tract to move mucus. Flagella, singular is flagellum. It means whip. Normally, a cell would only have one flagellum, and only one cell type has it, and that's sperm. So the flagellum is used for swimming. Microfilaments and microtubules are protein strands in the cell. Microtubules are found throughout the cell, and they build kind of a scaffold. They're made of a protein called tubulin, and they're basically little tubes. Tubule means little tube. Microtubule means little tube. They're the ones shown in purple here. So they hold the organelles in place and keep the cell's shape. So they're really the skeleton part of the cytoskeleton. Microfilaments, these are smaller. They're made of a protein called actin, and in most cells, they're just at the surface. They're shown here. They're used to change the shape of the cell. If it needs to, say, squeeze to move somewhere, if it needs to take in something from the surface, it needs to bend the membrane to do it. So the microfilaments are kind of the muscular system of the cell. Now, the microtubules have one additional function, which makes them like a cardiovascular system. Vesicles don't just swim where they're going. They actually have a protein on them that's kind of like little feet that walks along a microtubule. So your cytoskeleton overall is skeletal, muscular, and cardiovascular. So microtubules are a scaffold and a road. Microfilaments, like muscles. Then the last ones, intermediate filaments. They're called intermediate because they're bigger than microfilaments, but smaller than microtubules. There are a variety of types, and cells will have some types and not others, depending on what their job is. There's not really any that you can see on the model. On that one model that's different, I think it's got some of the microfilaments at least. Some examples of intermediate filaments include proteins that hold the nucleus's shape. So instead of microtubules, it has different scaffolding proteins. And keratin, which is a structural protein in your skin. Now, an additional structure that's on the composite cell photo and discussed in the lab book, but not so much in the textbook, is microvilli. Microvilli look a little bit like cilia, but they're shorter. They don't have proteins in them for movement. Villi means fingers. So they are little bumps sticking up from the cell. I think the bumps on here are meant to be microvilli. Their job is just to increase the surface area. The major place we have them is in the small intestine, where you digest and absorb the nutrients from your food. So that surface area gives more area for the food to touch and be taken in. Right. Any cytoskeleton questions? Our last thing is to look at the structure of the nucleus. Because the nucleus has the DNA, which is all the instructions for what the cell needs to do, it's often described as a control center or brain of the cell. This is Garfield under a blanket pretending to be an amoeba and saying, I have a nucleus egg. We mentioned that the nucleus has a double membrane. That's called the nuclear envelope. So that is 2 phospholipid bilayers. So where the cell membrane is 2 rows of phospholipids with tails together, double that to get the nuclear envelope. So you got 1 set here and then another set. So really 4 layers of phospholipids. There are holes called nuclear pores. To let things get in and out of the nucleus. RNA is a major thing that needs to cross. Inside the nucleus, all of this collectively is called nucleoplasm. There are 2 major things in it. A nucleolus, which is a dark spot, like ribosomes, it's made of RNA and protein. But where ribosomes build protein, the nucleolus builds ribosomes. And then the rest of the nucleus is filled with chromatin, which means colored stuff. Typically on a microscope slide, when it's been stained, it'll be easy to see the nucleus because the stain sticks to the chromatin. It makes the nucleus look darker. Even more stain sticks to the nucleolus, making it look even darker within the dark nucleus. Well, we won't always be able to see the nucleolus on our slides, but the nucleus usually stands out. Chromatin is the DNA plus some proteins to keep it organized. And it's just a mass in the nucleus most of the time, but it's made up of multiple separate pieces called chromosomes. Okay. So we have colored bodies that together make up colored stuff. So here's a diagram of the nucleus. So we have nuclear envelope and pores. They kind of look like little flowers because they're a little cluster of proteins that makes a ring that passes through the membrane or the envelope. Here's the nucleolus, and then all these other strands are chromatin. It's similar on the models. I actually like the model a little better because they didn't put the nucleolus right in the center. It can be anywhere in the nucleus. It's not like the core of the Earth where these diagrams often look like. It's somewhere in there. And then everything else is chromatin. And you can see little dents indicating the nuclear pores. And if you look really closely, you can even see that the nuclear envelope is double. That shows the phospholipid layers. This is a whole cell photo. This is taken with something called an electron microscope that picks up more detail. So the whole picture is the cell, then this is the nucleus, and all the scattered dark stuff is chromatin. The clustered dark part is the nucleolus. All right, so that's the end of what goes on the test.